Production of vaccines.
Vaccines are important biological products that prevent and control the occurrence of infectious disease. But how vaccine is made? Vaccine production are divided into following 6 steps. First step is selecting the strain for vaccine production. Second step is growing the microorganism. Both steps are upstream processing. The third step is isolating and purification of microorganism. Fourth step is inactivation of microorganism. The fifth step is formulation of vaccine, and the last step is quality control and lot release. These 4 steps are downstream processing.
The first step in vaccine production is the selection of strains. In the selection, the virus or bacteria must be free of impurities. This includes the similar virus or bacteria and even the variations of the same type of virus or bacteria. Besides, the strains should be obtained from recognized culture collection with established and documented provenance. The next step is the growing of microorganisms. The virus strain can be grown by using cell tissue culture, blood embryo, live animal inoculation, and transgenic animal. On the other hand, bacterial strain can be grown by using batch or continuous culture. Next, the isolation and purification of microorganisms. Isolation is the removal of components whose properties vary markedly from that of the desired product. Purification is the step to separate and retain the desired product at the highest purity. The first method of isolation and purification is centrifugation, which includes the differential centrifugation and density gradient centrifugation. The next method is filtration that involves gas unfiltration, tangential filtration, and ultrafiltration. Chromatography can also be used. The most commonly used chromatography are ion exchange chromatography and affinity chromatography.
Inactivation of microorganisms. Virus inactivation involves dismantling a virus' ability to infect cells without actually eliminating the virus. There are 2 ways to inactivate the viruses. First, by attacking the viral envelope or capsid. Second, by interrupting the viral DNA or RNA. Inactivation of virus can be done by using different techniques. First, solvent or detergent inactivation. Detergent treatment X100 typically used to disrupt the interaction between molecules in the lipid coat. This made the coat dysfunctional and impeding replication. Acidic pH inactivation or low pH treatment. Acidic condition deactivate virus. Solvent inactivation and acidic inactivation are most effective with lipid-coated viruses. Next is pulverization. Pulverization involves increasing the temperature of solution to denature the virus. These techniques are effective for both non-lipid and lipid-coated viruses. Next technique is ultraviolet inactivation. UV ray inducing the dimerization of nucleic acids. Thus, the virus cannot replicate their genetic material.
Then, the vaccines are formulated to enhance and stabilize the immune response towards the antigen. The main ingredients in vaccines are suspending fluids. They are sterile water and proteins which kill or weaken the microorganisms used in vaccines. Next, preservatives and stabilizers are added to ensure vaccine remain unchanged under heat like acid and humid. Inactivating agents inactivate toxins and kill unwanted bacteria or virus to prevent contamination. Then, adjuvants, primarily aluminum salts, act as enhancers to induce a stronger immune response. After formulation, vaccines are introduced into vials, tubes, or prefilled syringes for distribution. Before releasing the vaccines, they have to undergo quality control. The first test is sterility test, which ensures the absence of living microorganisms in the vaccine. Then, chemistry test check whether the product has the accurate amount of adjuvant and preservatives and accurate pH. Safety tests demonstrate that overdose of the vaccine is harmless. Residual toxicity tests verify the absence of harmful substance in the product, while efficacy tests justify that the antigen in the product meets the international guideline level. After a series of tests, the vaccines are ready to be labeled, packaged, and lot released. And there goes vaccine production. It takes around 6 to 36 months for the whole process, as vaccine manufacture is a complex journey.